Hi everyone, Assalamu alaikum. Assalamu alaikum. Thank you for giving me an opportunity to present my topic in International Brain and Biomedicine Conference 2023. Starting in the name of Allah, the most powerful and the creator of everything. My name is Arfa, Azeem, PhD student in the Department of Biotechnology and Genetic Engineering at Cohort University of Science and Technology. My topic of presentation is the whole exome sequencing identified a pathogenic FA2H variant in Pakistani family with hereditary spastic paraplegia. The contents of my presentation is the introduction, why we choose this study, objectives, material methods, result data, and references. First, I will give you a brief introduction of the hereditary spastic paraplegia. As we know that there are more than approximately 25,000 genes which are reported for the human and within the human body, out of which 10,000 genes are known to be reported for the brain morphology and physiology, which is a very huge part, which is responsible for the nervous system. And also we know that the nervous system is the most complicated system of the body, which controlling the all other systems of the body. And now if some mutations occur within these genes, so it will cause the neurological disorders, out of which hereditary spastic paraplegia is also the one of the neurological rarest neurological disorders, which having the phenotypes of poor coordination of hand, speech, and eye movement, and also the gait abnormalities, which is a major phenotype of the hereditary spastic paraplegia, and also there is a spasticity of muscles occur within the spastic paraplegic patients. Now, why we choose this study is we know that the neurological disorders are very and having the phenotype mixed phenotypes, and also there is a high clinical and genetic heterogeneity, which make it difficult to diagnose at clinical level. So we analyze it genetically to specifically diagnose this type of these type of disorders. And also the unavailability of cost-effective diagnostic feasibility, unestablished molecular diagnosis of the patients with hereditary spastic paraplegia in Pakistan. The fourth one consequences is there is a high rate of consanguinity, which is about 70% within the Pakistani family. So it causes to spread the such type of disorders within the Pakistani population. So to overcome these consequences, we develop some objectives, which is to identify a causative genetic variant within the causative genetic variant in patients with hereditary spastic paraplegia. To determine the disease-causing alleles, segregation in the family members of patients. Third one is to predict the pathogenic effects of genetic variant on protein structures and functions. To overcome these objectives, to achieve these objectives, we follow some methodology. First, we obtained ethical approval from the CUST Ethical Committee cohort at Cohort University of Science and Technology, and also we signed up the consent form from the affected family members. Second, we extracted genomic DNA and also direct sequencing. First, we prefer the direct sequencing of some common genes, which is responsible for the hereditary spastic paraplegia within the specific population. But if we didn't find by direct sequencing, so then we further goes to the advanced technologies like next generation sequencing, microarray genotyping, data analysis, and co-segregation of genetic variants within the families. Fourth one is to predict the pathogenicity of genetic variants and also mutant protein structure prediction within the affected family by the help of online available bioinformatics tools and software like Phi2, Enfold, Mutation Tester, etc. The results of this study is here you can see that the families consist of the 4th generation. Clinically, this was diagnosed as a hereditary spastic paraplegia phenotypes of this family member is the limb weakness occur in this in the affected member of the family, and also there is walking abnormalities and poor coordination. For the pedigree analysis, you can see the key at the right side of the slide. After the whole exome sequencing, we received the BCF file. Then it was converted by the online softwares to the annotated file. For further analysis, we applied these different types of filters on the annotated file. First one filter is applied for genotyping, and the second one is to identify the numeric frequency of the genetic variant. Third one is for coding effect, in which we selected missense plus frame shift. The fourth one is for applied variant location effect, in which we mostly choose the exonic plus splice site region. Fifth one filter is applied for the HGMD phenotype, which is the hereditary spastic paraplegic phenotype. Finally, we find out the identified a FA2H mutation in FA2H, which is the deletion of codons occur from the 159 nucleotide to 167. There is a deletion of nucleotides occurs, which affected the protein structures and protein at the position of arginine 53. Codon 5 codons were deleted, so it affected the protein structures and also the function of the protein. So for further analysis, in silico analysis was performed, in which we use different online bioinformatics tools like the mutation tester, and also we use the Versome, and for the identification of the numeric frequency, we also use the Genomed software. Here is the summary of the this presentation. First, we located the family, then we signed up the consent form, and also obtained the ethical approval for this study. Then we analyzed the family, genetically analyzed the family. So we find out the FA2H genetic variant and the FA2H gene, in which deletion of many nucleotides occur, and also which affected the protein and structures and functions, which is protein structures and functions. Also, you can see that the complete segregation of this variant within this family. Here you can see that the mother of this patient is heterozygous for this genetic variant, and also the one sister is a heterozygous carrier for this genetic variant. One brother is a heterozygous carrier for this variant. The two sisters, the first one and second one sisters, are normal for this genetic variant. So here is the differences of this study. Thank you for questions. You can contact me with this email, through this email. Thank you so much.